Hi, and welcome back to the BioHackers podcast. I'm your host, David James Clark IV, and I'm joined today by the esteemed Mr. Dr. Alex Feltus. How you doing today, Alex?
Yeah, I'm all right.
Yeah.
No, I'm doing great, David. How are you doing?
You look fabulous. Are you ready to talk bugs?
I am, and this is our guest is in India, so it's very late where I am, and I had a cup of coffee, so let's see what happens.
Oh, this is going to be so cool. Cue the opening song. All right. All right. So today, as you just said, our guest is coming from the other side of the world, and we're very, very excited to hear about his research and the use of genomes and AI and biochips in bugs and pheromones to help crop production, clean food. Tell me a little bit about this crazy idea, because it sounds too good to be true.
Yeah, I mean, I know a little bit about it from what the speaker is or your guest has done, and I'm excited to learn more about it myself. But this is an attempt to, I guess we'll let him speak for it, but attempt to try to do things in a more natural way to bypass crop damage through pests, and we're going to need faster solutions as climate change occurs and bio-based solutions to be able to feed the people, right? So he's a biohacker, even if he's not a computational biologist, and I can't wait to hear what he has to say.
Well, and he's solving a huge problem. I mean,
Yeah.
A few podcasts ago we talked about Jurassic Park, and and the genetic manipulation happening there. If you've seen the latest installment of that storied franchise, they cross dino DNA with locusts and basically ruin all the agriculture in the world. So,
wait, spoiler alert.
I know, spoiler alert. So you have giant dino locusts, but it really got me thinking about, the importance of agriculture and pesticides. And then you go to the grocery store and of course, if you're interested, you can pay twice as much for organic food. Which again is food that hasn't been spoiled by any of these poison-based pesticides. So it'll be really interesting to hear what Markandeya has to say about the use of natural technologies, natural methodologies around pheromones and bugs. You know him. I mean, he's kind of a self-proclaimed, chemical engineer, biochemist, geneticist, but you know him back from more kind of traditional genetics. Is that right?
Really, genomics. He was back, it's been probably 2 decades now, last time when I worked with him. But he was working on microarrays, which are little tiny chips that you can print DNA molecules on, like physically print them, like with semiconductor technology.
Wow.
And then you can actually measure how much of a sample, how much DNA or RNA binds to the chip. So these were like things where this is back when we were doing them on slides, and he was printing them with a machine that would have pins that would go grab DNA from test tubes and print it onto a little slide. So there wasn't even like any like real modern tech. I mean, it's pretty serious tech back then, but this is the beginning of genomics when it was really hitting hard right after the human genome was sequenced. He was doing this, and he was doing it in rice plants. Oof. And so he was like really big, bringing huge genomics to crops. He's been doing this for decades now. So this is another iteration.
Well, and we were kind of going back and forth preparing, and I was doing a little bit of research preparing for the episode. He used this term biochips, so I'm going to be asking him about that. But is that a little bit of what you're talking about?
We'll have to see the context of how he's using biochips. That's one of those things that can be used for lots of different reasons or lots of different applications. So we'll see what his tech is.
All right. Well, I think that's a really great segue. Let's welcome Dr. Markandeya Gorantla to the show. Here we are with Dr. Markandeya Gorantla. Thank you so much for joining the show. How are you doing today?
I'm doing good, David.
Wonderful, wonderful. Thank you so much for joining the BioHackers podcast. We're really excited to talk about insect pheromones and your work in agriculture. Alex, why don't you give us a quick introduction to Dr. Gorantla and how you two met, and we'll get a little bit more comfortable with his background.
Yeah, definitely. So Markandeya is, I met him, I guess it's been 20 years, maybe 2003, something like that. It's been so long, it's not even sure. But so I've kept touch with him through LinkedIn and things over the years, and he's now the founder and executive chairman of ATGC Biotech. Make sure I don't get that acronym's used for lots of things, right? From DNA.
Yeah.
But so he's been doing fantastic work over the years, and always been intrigued what he's doing, and so excited to learn more about the tech that you have and how it can be used to help feed us, right? Which is what we were doing together, I guess, 20 years ago. You were building microarrays. You were doing it. I wasn't doing anything. I was just there to help you out and be in awe at your molecular biology skills. You're still the best molecular biologist I've ever met. I can't believe how clean your work is.
Thank you, Alex.
Yeah.
Excellent.
So, so let's start there. I mean, I'm always fascinated to hear a little bit about Alex's past. I don't think I've ever gone back 20 years. So this is a unique opportunity for me and for all the listeners to get, insight into the Alex Feltus circa 2000. So Markandeya, how did you work with Alex and what was it like back then?
So back then I was on deputation from Rockefeller Foundation. So we were working to save water. So India produces a lot of rice, and the idea was to identify drought tolerant genes, and Alex helped me during that process. India was not having the equipment which is required for the studies. So I requested Alex to help me out. So he was my mentor at that point of time. So I went to him, learned, and great help from him. We could fingerprint all the genes which were responsible on the chromosomes, and then today that has become a reference point for many of the breeders to breed for drought tolerance rice cultivars.
Yeah. That was an exciting time. That was, that was really, the human genome had just been put online, I guess, for a few years, and the work in rice and other plants or crop species was brand new. We didn't have genomes and had very, very limited resources. So people like Markandeya was building microarray chips and helping the breeders. It was an exciting time. Everything was just really shifting over from humans into plants, and it's never stopped since then.
It never stopped since then.
Yeah.
So in the interest of, learning more about our biohackers, and you definitely qualify, tell us something surprising or interesting about yourself.
I shifted from one field to another field. So from plant biology to oncology, and from oncology I learned, again back to plant biology. The main reason of shift because, when I was working in oncology, all the incidence of cancer was correlated to pesticides. And and I thought, we should develop a very safer molecules to provide a cleaner food. So when I looked at it, I think the industry has been struggling to develop even 1-2 molecules per year with billions of dollars. So what could be the alternative was my thought process. At this point of time, I thought insect chemical behavioral modification to confuse males to achieve an kind of an insect family planning program in the field.
Insect family planning.
I love it.
So I thought this is the only way to tame the insects. There is no other way to control their population. So this excited me, and I started developing tools to manage the insect population by using pheromones.
What was it that gave you this idea? Was it just completely your idea? Did it come out of the air, or did you read about it? Like, that's such a unique way of doing it, solving the problem.
Yeah, I was exploring for all the molecules, biologicals, biorationals, and the new molecules for the insect control. But looked like all of them had its own challenges, and the pheromones was the only one which was sustainable for last 40-50 years. We have never seen any resistance.
Really?
So, yeah. So because if you see any resistance, the species will end, isn't it?
Yeah.
So that's the beauty of it. So the only communication between a male and female for their progeny to flourish is mating. If they develop resistance against mating, so the species itself will vanish. So that's the reason why there is no resistance for this particular mechanism of action. We are playing with the mind of an insect. We are modifying its behavioral response.
Mm-hmm. And so is it a reduction in crop damage, or is it an elimination of crop damage relative to traditional insecticides?
Yeah, traditional insecticides, we chase insects,
Yep.
Here we tame insects, that's the difference. So by taming insects, you are not asking them to breed. Insects breed in millions.
Yeah.
So one insect in entire crop cycle, so they transform into 1 million. 2 moths, 3 moths transform into 1 million. So that is kind of because each of the female lay more than 200 to 1,000 eggs.
Right.
So, yeah, so the multiplication is so high. They have 30-day cycle of each of the life cycle. And in 4 broods, you see millions. So the idea is the damage to the plant occurs when there is above economic threshold level. So the economic threshold level happens when in second brood or third brood. So the idea here is to manage the first brood itself, which can be through using sex pheromones or aggregation pheromones or many of those things, which change the insect behavior pattern. And that's the way you do not allow the male to find its female partner, and you achieve a kind of mating disruption. So that's how it is.
Is there much diversity in the pheromones from a particular, like, actually what's one pest that you're focused on right now, and maybe we could talk about that as a scenario.
Yeah, so basically insect epidemics. So we are experts in managing insect epidemics.
Yep.
So you might have heard locust migration, isn't it? Mm-hmm. So locusts, they damage entire field in few hours. They eat away the entire fields. So these kind of epidemics keep coming now and then because of the environmental change, climate change. And in agriculture, so fall armyworm is one of the greatest epidemics people have seen, especially in Asia Pacific and Africa, when the fall armyworm from Americas came to Africa and then to Europe. There was entire destruction. I mean, it was leading to famine.
Yeah.
So that was a kind of destruction between fall armyworm. And here we see the of pink bollworm. So the BT cotton was giving very good resistance against this, and later off late, the moth became adapted to the BT toxin levels in the plant.
Right.
And today it's an epidemic. So insect epidemics. So insect epidemics happen when there is no solution, there is no control. So insect epidemics can be managed through this particular technology. So we are experts in area-wide management. So we work with various governments, US federal programs, and in India and Africa. So we work with the governments and we spray where the pupa come out of the soil. So when they come out of the soil, when you spray in lakhs of acres, the male and female will never be able to mate and there will be no egg laying. Even if there is egg laying, the eggs are sterile.
Right.
So, so commercially, we are not yet in the market. I think in last 2 years, this space has got a lot of attention. So we have been using this technology to address epidemics, insect epidemics, basically,
Right. So with like a locust, for example, maybe, what, if you're familiar with one particular instance of that. So is there one pheromone that you need to be able to stop reproduction?
Exactly.
Or you say there's no resistance, but is there much diversity from different broods, producing different pheromones?
Yeah. Each insect produces its own pheromone. So it's like our fingerprints. So each species has its own pheromone ratio and blend, and all the pheromones are different for different insects. And sensitivity also is different. So few of them, they get attracted to almost 1 kilometer. Okay, that is the sensitivity. And few of them, they need very high dose of pheromone. They are less attractive and they are attractive from about 100 meters. So specificity is very, very clear. So it's like a fingerprint. So each product is so species specific. That's the reason I say, when we use this technology in agriculture especially, you are targeting your species of interest. Means we are providing farmers a choice to choose which pest he needs to manage. Unlike today, all the available insecticides are basically broad spectrum. They kill all the predators, including predators, honeybees, etc. But however, this technology will only focus on managing the species of your interest.
Yeah,
That's really cool. And so just like for the people that are out there, I mean, like this all stuff we talk about on this podcast is always really hard. It's hard for me and everybody to understand. But basically this is analogous to like antibiotic resistance, right? I mean, it's the same level of a problem, I guess. But you just they don't get resistant. That's just really amazing. That's cool. The specificity is that high. So do these work? I mean, you're doing trials across the planet, right?
Yeah.
Aren't you doing
across
it
the
in multiple
world?
countries right
across
now?
the world. We have done in Argentina, US, Africa, Australia, India. In India, we have developed a manual application like a toothpaste. Here people call as a toothpaste. Whatever they apply on their brush, they apply onto the plant. They create 400 artificial females by putting our product into the field. And these are, then these 400 droplets when they apply per acre. So the male will go towards these droplets, not to their virgin females. And in other countries, in US and Australia, we do aerial applications in lakhs of acres.
Right. Wow, that's amazing. That's just absolutely amazing that you do that. And so they typically work in different countries then, right? I mean, you're the same. I'm sure in science there's always outliers, right? There's some things, there are experiments that don't work, but is this something that, I mean, you're obviously doing very well with your company, but is this something that is, I mean, I don't think it's not very common knowledge, right? At least in the U.S. to talk about this form of control. Is it becoming bigger? I mean, is the market getting bigger? Is it becoming more, applied?
See, the entire damage because of insect is almost like about $570 billion. So out of $570 billion today, hardly 30 to $40 billion is managed by the GMOs with the BT toxin and the insecticides. So insecticides contribute around $20 billion, close to $20 billion, and the GMOs contribute to $32 billion in all. So this is the only thing which presently we have solutions. And the rest, almost like about $450 billion loss, we are not able to manage. The reason being, they are internal feeders and borers. Many of the pests, they live inside the plant. Okay, so as soon as the plant germinate, they enter through roots and they live inside the plant, where conventional solutions are very, very tough to manage them. And they, in the net, the vigor of the plant, which has to produce double, is not so. What we today, the farmer has never seen the actual yield of the plant, of actual yield potential of the plant. If you are able to manage these pests, which are internal feeders and borers, you will get additional $470 billion food to feed.
Right.
Okay. So that's where we are working. Wow. So we are working for the unmet need of the agriculture. The space will grow. Will grow at least 10 times than the present both the insecticides and the GMO industry. So there is a potential of more than that. The only limitation is the building blocks of these pheromones, synthesis of pheromones. It was a very tough challenge. It's a big challenge wherein we worked for the last 15 years to build these building blocks.
Have you done that in your company, or did you partner with chemical companies to do that?
Yeah.
How did you do that?
That was a typical challenge. So when we did the initial survey, when we began in 2006 and 2007, when we did the initial survey, each of the company there are 5 elements, like, the building blocks to build a pheromone, and the building blocks will result in intermediates, and intermediates will result in the pheromone. After pheromone, you have to formulate them in a delivery system which delivers the pheromone across the season in a linear zero-order release. Because these pheromones are so sensitive, it works in picomoles. So industry was using almost, the industry was almost using 150 grams, which really was not required. So you can as well manage with 5 grams. Okay, so that is the where we worked on. So we worked in-house. To develop all the technologies, right from the building blocks to the final delivery system. And today we are very proud that we have 5 different delivery systems for pheromones: a tablet, a spray, a paste, and an emulsion system, which can give season-long control with a linear release. So that's where our specialization is. Yeah.
This is where I guess your diverse past is really important, right? I mean, you're solving is an interdisciplinary problem at an industrial scale. It's amazing that you're able to do it at all, let alone be as successful as you are.
Yeah.
Fantastic. So I know in some earlier in some of the communication we had preparing for the episode, you talked about yourself as the Pied Piper of insects, and I love that concept where you're using this pheromone to generate behavior modification. So can you help us understand a little bit more the genetics or the genomics or the science behind, was it biochemistry that allowed you to create those building blocks? Was it genetics or some sort of AI biochips that helped drive the delivery mechanism? What, without giving away any corporate secrets, help us a little bit understand the science of what you've done, because it's really fascinating.
Yeah, I think every science which I learned, I think it helped to build this industry. Especially my chemical engineering helped to scale up. The main limitation for this technology is scale. So the industry was not at all growing because of the chemistry, which is not scalable. So first thing what we did is we did a scalable chemistry. So the chemistry was scaled up. Today we produce almost like 20 metric tons of these pheromones. Our country, country imports only 15 kilograms, whereas we export almost 20 metric tons for 75 different pests. So today, whenever I go to anywhere, the male insects keep following, and they land on my hair, they land on my shirt, they land on my nails.
So you use it as cologne.
It's like so that's a problem I have. So wherever, even including when I sit in a lounge, the meal moths, they come only to me, not to any others, because.
And they try and mate with your fingers. That must be an interesting experience.
Yeah, that's what they do. So I can share one photograph of my.
I'd love to see that.
a male landing on my finger and doing something.
Oh, that's great.
So these are the things.
So
and that's the first part about scale. So we were for the last 15 years we were able to scale up. And second part is my genomics, how my genomics knowledge we used for building the pheromones. So what we thought is it takes 6 months to synthesize chemically. It's the same biosimilar which basically is released by insect, but to synthesize that it takes 6 months. So how do we reduce? So we have to address this $470 billion loss due to insect pests, and how do we address such a large volume production? So then we thought insect synthesizes naturally. Okay, so there might be a biosynthetic pathway which basically the insect uses to produce pheromones. So our knowledge in genomics, with few of our tricks, with few of our algorithms, we did the transcriptome profiling of the male and the female insect olfactory glands, and this revealed the actual genes which are responsible in that particular species, how the pheromone is naturally produced. Now today, so today we developed, we identified those genes, we cloned them in yeast, and the yeast we use them as an immobilized cells to transform oil to pheromones. All we need to do is put oil, use the immobilized cells which contains the genes of the insect, and in 48 hours you produce the pheromone. And the industry is producing, 1 or 2 companies are producing globally, but what they are producing is they borrowed genes from other species and feeding the substrate which can produce the pheromone of their interest. But that should not be the case. So how we, with our knowledge, with our genomics knowledge, what we did is we identified species-specific biosynthetic pathways. So that is the novelty of our company. So the species-specific biosynthetic pathways will produce species-specific pheromones. So this is the very new. I think this technology will revolutionize how we manage the insect pests, because you are producing the species-specific pheromone through biosynthetic pathway of each of these species, and when you deploy in the field, you are managing that particular species. So a farmer has a choice. So in the future, by maybe another 3 to 4 years, when a farmer goes to an insecticide shop, he has a choice which pest to manage, how to manage.
And to do it naturally. So earlier, Alex and I were talking about the huge growth of organic food and how, people are becoming more sensitive to what's being put in their food. Is this something then that people can think of as a fully natural solution, and none of the food, you talked about clean food. Is that really the byproduct of this process?
Exactly. That's what it will lead to. So we are rebuilding the ecosystem without any chemicals. So this particular product, this particular intervention will really not require any insecticide application. And we are not initially focusing on the existing problems. There are several larger problems. The problem is tenfold. So the population is growing.
Yes.
The population growing, land is limited, and we need to produce more. And the loss where there are no solutions is what we are presently addressing. So whatever we are addressing will lead to organic produce, clean food for our future, to feed the future. And that's where we are now.
I guess at those levels that their effectiveness is so small that, can you even detect these in food, right? The levels must be almost impossible to detect.
So
right?
Pheromones, though it should not be taken as a pest control, it's an insecticide. So it's basically we are trying to change its behavior. So in general, we easily metabolize pheromones. In nature, millions of insects release lots and lots of pheromones naturally in the air. So the air we breathe has almost 10 to 20 times than what we apply in the field. And here, so our body easily metabolizes this pheromone. So why, when we are breathing 10 to 15 times the air which is saturated with pheromones, how does the technology work in the field? So what we apply is species specific. So what we breathe is contaminated with millions of insects. Okay, so that's the difference. So technically there will be no residues. There will be no effects on any other organism or honeybees. So it's very, very safe.
And it's safe, and it works when things are resistant. If there's no solutions, right? And there's just going to be more and more of that, right? I remember back when I was working with you a long time ago. I was trained as an oncologist myself, right? I think we reversed our paths a little bit, right? I'm still doing a little bit of both. But I remember that the climate change was of course been a topic since I was a kid, but it's really picking up a lot of steam. And I was watching, looking for evidence and way of explaining what this is happening to people, to myself, but also to colleagues. and friends. And I was noticing the United States Department of Agriculture, the USDA, they have a growing map, grow zone map, where they would show the different like times to plant different crops, right? And then because the temperature was already rising, this is back in 20 years ago. It was really that's when it's really starting to pick up steam. That the maps were moving north. Like you could look at the 1998 map and then look at the 2003 map and see it moving. So I presume that you guys could do that too, right? You could be predicting with the temperature change that's going to make pests move their zones, right, of where they reproduce. So you could actually have the guns ready for the battle against the pests as they move further north and south. Right. And
this movement also, we are actually developing novel tools. So when we talk about managing the mating, we also need to manage the population. So the easiest way when we are trying to identify the biosynthetic pathway of pheromone production, we also identified the male receptors. So the male receptors are the ones which actually dock. It's a protein. The male receptors are protein. So they dock the plumes of the pheromone, which is released by the female insect, and each docking has its has a note which says there is a female. So this particular aspect of this immunochemical docking to the protein. So we are using that, identifying those genes, making them into biochips. Now these biochips, when you place in the environment, in the field or on the roads, so you will be able to tell. We are writing algorithms, so to understand. So the biochip will read how many females are there in the field. Okay. Yeah. So as the females migrate, so the biochips will sense. And through artificial intelligence, they will identify how many number of females are there. Okay, so the movement of the migration of the insect pest can be easily monitored, and thereby the predictions of what to produce, where to produce. So if the moths are migrating in one direction, you produce in this particular geography. Okay, and when they are producing north, you produce in the left. So that way you can easily manage what to produce when and where.
So brilliant. That's fantastic. I didn't realize the power of that. And you can't. There's no way of doing that with traditional pesticides, right? I mean, you wouldn't be able to. You could still use that as a guide, I guess, but it doesn't really make a lot of sense to do it that way with what you're doing. It does make a lot of sense. Wow, that really helps with mitigation and just planning. Planning this.
So it was actually a junk in our entire research. So because we were focusing only to identify what is there in the female and things like that, but and when we subtracted the male receptors with the female receptors, so that's where you have few genes which basically are male sensory genes. So we thought it is a junk, but later we realized those can be used to convert to biochips to identify the females.
Excellent. So I just love your this interdisciplinary thing. So what we're doing with this BioHackers, so we're doing a lot of hackathons. We're bringing people together, the computer scientists, very computational, the geneticists, all these different people, and trying to come up with solutions by whatever means necessary, right? And we're, a lot of AI and stuff, a lot of data science is driving a lot of this now too. And also big compute. We have to have big computers to be able to do a lot of the genomics work, right? But you're like the perfect example of somebody, of what you can do. Like nobody could ever predict when we were working together 20 years ago where you would be now, right? But mixing together these ideas, being creative, and then you're actually able to turn that into profitability, right? I mean, you're helping the planet, and you're helping, people with jobs and everything. It's just that you're the best molecular biologist I've ever met, and you have another a place for me to put you on for
another pedestal.
Another pedestal. That's fantastic that you're pulling it off. But I just love it. Because I've learned over the years, and you're a perfect example. I'm sure you've seen this too. When you put together people that think differently, and then there's a common goal, a common problem to solve. Magic can happen, right? Absolute magic.
One of the themes that we've had in our episodes in the BioHacker podcast has been around the interdisciplinary element of science, but then also this concept of wet lab and dry lab, and in silico is a cool term that I like that Alex uses. So there's clearly an element of wet lab. You've done some sequencing and some biochemistry, but then also some computational biology, I would imagine, right? As you start to develop that. So can you talk a little bit about, which one is more? Are they equal? What role did computational biology play in the development of these, these pheromone components? I mean, we're always very fascinated to see how the science is interdisciplinary.
Yeah, so computational contributes 30 to 40%, I should say. So yeah,
great.
So unless we need to find a lead. So the process is 10 years in general to map out the pathways. If you are not going in a proper direction, so you end up nowhere. So computational biology really helps you to guide in a proper way. So biology, to complete the entire research, the program, I think it's 5 to 8 years. Any biotech, the wet lab is research is it's minimum 3 to 5 years, not less than that. And we are to be very sure that we are going in the right direction. And computational biology really helps. The algorithms really help, and that's how the success rate is. And computational biology, the teams are very, very limited. I think many of them, they go to the other software. Computational biology is a subject which more and more people should adopt to help other researchers. So we have today 90% researchers and 10% computational biologists. But the contribution is,
okay. Well, I think I just saw an opportunity for the biohackers because the audience of folks that we're certifying here at Praxis are almost all computational biologists, and if you said 30 to 40% of the work is computational biology, but only 10% of the people have those skills, there's a gap there. There's a skills gap, and that's something we've recognized just over the last few years. And one of the reasons, find a need, fill the need, that we came out with the BioHacker certification program, so people could get trained remotely, they could do research through the biohackathons, and then they could get certified with digital badges in things like bioinformatics and plant genomics and computational biology. So that's music to our ears, and to all our listeners. Go check out Markandeya's company, and it sounds like he needs some more computational biologists. So did the computational biology then help you decrease the amount of time? Did it save you time on the regular biology? Did it help you scale faster? What kind of advantage did it give you?
It scales almost, the scale up is almost, it brings down 10 years of our time.
Oh my goodness. Wow.
Yeah.
Oh, that's fantastic.
That's it.
Okay, so that helped you tremendously.
Correct.
And we all know time is money, so 10 years is a lot of money. And I think, Alex, that's what we're seeing in our biohackathons as well. Is you can run a really impressive experiment. It might take you a few weeks or maybe a month, but you end up with results that either close a door or open a door, and that's that's research that may have taken a year or so, in the old-fashioned way. So the ability to run multiple models. We had a wonderful Clemson professor on the show recently who's built some really impressive mathematical models for modeling biological systems and cell systems, and being able to again run simulations, very, very sophisticated advanced simulations, for the purposes of speeding up and scaling research. So I think very creative people doing interesting things with supercomputers to help again, speed up and scale research.
True.
Yeah, what will happen, right? I mean, I see this computational biology. I mean, the wet bench biology is critical, right? I mean, to do it that way. But I don't know what's going to happen with this, and I see the companies. I really started the training that I would do with my PhD students and undergrad students too. The companies were hiring them even at the undergraduate level, and then when I would talk to them, they would say, "Do you have any more people?" They're, people probably like you that are always looking for new talent, right? And there isn't a lot of talent out there, and that's really the driver of what we're trying to do, because I think that fuel, that data science, understanding biology, that mixture of skills that is in a computational biologist, I don't know how much time it's going to save lots of time, and it's going to develop products or reduce product development cycles and breeding cycles for new crops, right? But also just the ideas that we haven't even come up with, kind of like what you're doing, just incredibly creative and novel ideas that will come out of this, right? I just wonder what's going to happen for our species if we, have 1,000 times more computational biologists out there, right? What will they do with that knowledge? I'm excited about it.
Yeah, I'm excited about the future with people like Markandeya, out there applying these novel and creative application solutions to very serious problems, and that's food shortage and insect epidemics. So we're starting to get towards the end of the show, and I always like to wrap it up with one last question. We talk about biohackers, and the question is for you, Markandeya, what is a biohacker to you?
I think it's good to know that people get certification. They get an opportunity to learn computational biology through workshops and through this particular forums. I think it's a good tool, Alex. I think the number of students you are teaching now, it gets expanded to the society.
Yep.
Okay, the society will learn, and it's a direct transformation from your lab to society. We call from lab to field.
Definitely.
So Alex is doing from his computers to society, translating the computational biology to the society.
Right. I think that's so important, right? To always have a focus of something that's beneficial to society, but also just beneficial to commerce too, right? Or discovery. But yeah, just a little bit less. I've always found myself a little bit less basic science, a little more applied, is so much more rewarding for me. So.
Well, thank you so much. Do you have any final words for us, Markandeya? Again, we're very impressed and appreciative to have you on the show today. Any final words?
Thank you. It gave an opportunity to meet my friend Alex. 20 years. I'm still depending on with computational biologists like him. I don't have that knowledge.
Yes, and the genomics that he taught you 20 years ago, you're still putting to very good use. So. Thank you, Alex. Thank you, Dr. Markandeya Gorantla, for joining us today. And that wraps the show. Take it to the closing song.